All right, can somebody confirm you can hear me?
I can hear you.
Great, thank you. Let's see what I can close down here. I have the new syllabus online. I'll show you in a minute. Let me call up the syllabus now. Oops. I don't know, are we on chapter 5 or chapter 6? Should be on chapter 5. No, that's Tuesdays, Thursdays. Let me back up a bit. Oh, there we are. Found it by accident. All right. All right, before we begin, are there any questions? Let me share my screen. And where am I? Right here. So I've uploaded the new schedule, so you should be able to find it in the syllabus. Let's let this load, hopefully. Well, it doesn't look like it's loading. Well, oh, there it goes. Sort of. So about page 10. Must be page 11. The main change in the schedule is the quiz scheduled for today has been postponed until, why the heck isn't that on page 11? Until next Wednesday. Any questions about that? That did require me to move down quiz 4 because I didn't want you to have two quizzes on 1 week. But I decided to do that because I haven't gotten lab 6 graded. And to tell you the truth, I'm still grading lab 5 for my Tuesday, Thursday people. So I'm behind schedule. All right, any questions on anything? If not, let's take a look at the schedule. Today is February 14th. And is today Valentine's Day? Nobody's going to comment? All right. Happy Valentine's Day if it is. Uh, we're supposed to be covering chapter 5 and finishing chapter 5 today. I don't know if we'll finish chapter 5, but we'll try. There is no lab for tonight. You're supposed to be working on your unknown project. I should also point out to you your infectious disease paper is coming up. It will be due Saturday, February 24th. So that's, well, 1 week from this Saturday. Any questions about any of that? I will show up in the lab just to answer questions. So I will be there at the normal time and I'll be there until 15 minutes later. But if no students answer or show up, after 15 minutes, I will leave. So I'll be there only for the first 15 minutes. The exception would be if there's lots of students who have questions, I will continue until the last question is answered. Any questions on anything before we begin? All right, let's go to chapter 5. Carbohydrate catabolism was the last topic we discussed. Oh, I need to show you the video. So let me do that now. The video on aerobic respiration. Come on. After the
This is
commercial.
Dave. Had a tight deadline.
All cells need energy to stay alive. Usually your cells get their energy through the process called aerobic respiration. The word aerobic refers to oxygen. In aerobic respiration, oxygen interacts chemically with a molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into 2 molecules of pyruvate, energizes 2 electron carrier molecules, and produces 2 molecules of ATP. The next 2 stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create 2 more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
All right, any questions about that? Yeah, so that's an overview of aerobic respiration. Let's talk about aerobic respiration in more detail now. I'll pull this slide up. Here we're looking at aerobic respiration, comparing it to fermentation. Aerobic respiration has 3 principal stages: glycolysis, the preparatory step in the Krebs cycle, and then lastly, the electron transport chain. Essentially, electrons are flowing from glucose, an energy-rich molecule, to carbon dioxide and water, which are energy-poor molecules. There's a high yield of ATP from aerobic respiration. Now, fermentation starts with the same. You have glucose, which enters glycolysis, the first step of aerobic respiration. But there is no Krebs cycle, there is no electron transport chain. Instead, the pyruvate made by glycolysis is converted into a fermentation product, such as ethanol or lactate. There's a much lower yield of ATP in the fermentation. Any questions about any of that? This is showing you an overview of aerobic respiration in a eukaryote cell. So if we put the pink right here, a cell, like a square cell, sort of like a plant cell. Glycolysis happens in the cytoplasm of the cell. Glucose is converted into pyruvate. There is some ATP that's made, some NADH that's made. We'll talk more about the NADH in a little bit. The pyruvate in a eukaryote cell then moves from the cytoplasm into the mitochondria. inside the matrix of the mitochondria. This is where the preparatory step and the Krebs cycle happen. Here it's called the citric acid cycle, and we'll talk a little bit about that. The Krebs cycle has at least 3 different names. Citric acid cycle is one because there's a lot of citric acids in the cycle. It's called the Krebs cycle after Dr. Krebs, who discovered it. Your textbook and I tend to use the name the Krebs cycle. In the Krebs cycle, some ATP is made right here. And this is actually showing you the preparatory step right there. In the preparatory step in the Krebs cycle, CO2 is made. NADH and FADH2 are also made. The NADH and the FADH2 from the Krebs cycle and the glycolysis carry the electrons from glucose, or the molecule derived from glucose, to the electron transport chain, which is right there. It does have another name. I don't use that name. And this happens, the electron transport chain, across the wall of the inner, actually the membrane, across the membrane of the inner mitochondrial membrane in eukaryote cells. There's a high yield of ATP being produced by the electron transport chain. Now that's discussing it for a eukaryote, which have a mitochondria. In prokaryotes, the glycolysis is happening in the cytoplasm. The Krebs cycle and the preparatory step are happening in the cytoplasm. The electron transport chain is happening across the cell membrane. Any question about any of that? All right, so the first step of glycolysis, of aerobic respiration, is glycolysis. It does have another name, which I do not use, Emden-Meyerhof pathway, and I'm not German, so I could be butchering those names. In glycolysis, glucose is oxidized to form pyruvic acid. Glucose is a 6-carbon molecules. Pyruvic acid is a 3-carbon molecule. And so 2 pyruvic acids are made per molecule of glucose. Glycolysis occurs in most living cells. It can occur in the presence or the absence of oxygen. It does not need oxygen, but it can work in the presence of either oxygen or not. It does produce some ATP and NADH. Any questions about glycolysis? Let me briefly mention this about pyruvic acid. Pyruvic acid is essentially the same thing as pyruvate. You need a chemist to distinguish the two. The pyruvic acid has the hydrogen attached to it. The pyruvate has the hydrogen lost. And the cell treats pyruvic acid and pyruvate the same. So I will use those two terms interchangeably. And like I said, the chemist wouldn't like that. But for our cells, it can use both pyruvate or pyruvic acid the same. Any question about any of that? All right, here we're seeing glycolysis, seeing all the steps, starting from the preparatory. Never mind, we won't call that the preparatory step because that's the preparatory step of glycolysis. It's not the preparatory step of aerobic respiration, so I won't even talk about it. But in this chemical reaction of glycolysis, we have a number of biochemical reactions occurring. 1, 2, 3, 4 going here where we make either this one or that one, and that's a 6-carbon molecule. It breaks down into 2-carbon molecules. Not two, 3-carbon molecules. This one continues in glycolysis. Come on, mouse. all the way down. That one, if you notice, will be converted into this one. And then we will go down glycolysis until the very end, you end up at pyruvic acid. Along the way, 2 ATP come into glycolysis. That's here and here. And then 4 ATP come out, 2 there and 2 there. That means that in glycolysis, we get a net gain of 2 ATP. Any question about any of that? All right. Well, when I was an undergraduate, I had to learn all of the intermediates of glycolysis and all of the enzymes making the biochemical reactions. Since I had to know all of this, I thought it would be fair that I require you guys to know every step of glycolysis. What do you guys think about that? Everyone's okay with it?
I feel like I'm glad we don't have to know all that.
So you looked at this lesson ahead and you know that I'm not going to require that.
Yeah,
But
thank you.
I do require you to know the summary of glycolysis. You start with glucose. You get a net gain from glycolysis of 2 ATP. 2 pyruvates come out of the glucose, and you can call that pyruvic acid. And then 2 NADH come out of glycolysis. Any question about any of that? All right. Before I go on and talk a little bit about aerobic respiration, let me state that respiration is the oxidation of glucose or some other molecule which releases electrons for the electron transport chain. And so in respiration, we're catabolizing mainly glucose, but you can catabolize really any molecule of food that you eat. We'll talk a little bit about that near the end of the lesson. But there are 2 types of respiration. There's aerobic respiration, where the electrons from glucose will end up at oxygen. So we say the final electron acceptor is oxygen. And that's different from anaerobic respiration, where the electrons flowing from glucose will not go to oxygen. They will go to some other molecule. And these we call anaerobic respiration. Along the way, ATP is generated by a respiration, regardless of whether it's aerobic respiration or anaerobic respiration. Any questions about any of that? Just a minute here. I feel like I'm going to be sneezing. Tried to blow my nose, see if that would help, but that didn't seem to help. Not sure why I'm feeling the urge to sneeze. Hopefully I won't sneeze in the middle of the talking here. So after glycolysis, we then enter the preparatory step. In the preparatory step, the pyruvic acid is converted into a molecule to enter the Krebs cycle. So pyruvic acid itself cannot enter the Krebs cycle. It has to be converted into acetyl coenzyme A. In the process, we do generate 1 NADH molecule per molecule of pyruvic acid. And then coenzyme A comes on. One carbon comes off the pyruvic acid, so we have the first carbon dioxide molecule made from glucose. You should note that the preparatory step runs twice for each molecule of glucose because glucose will be divided into 2 pyruvic acids. So the preparatory step will run twice. Any question about any of that? All right. So acetyl coenzyme A can then enter the Krebs cycle. The Krebs cycle has a number of different names. It's called the tricarboxylic acid cycle, or TCA for tricarboxylic carboxylic acid. So T, come on, mouse, T, C, A. It's called that because it has a lot of tricarboxylic acids in it. It's also called the citric acid cycle because citric acid is the first molecule made in the Krebs cycle. And this is abbreviated TCA for T for the, C for citric, and then A for acid. I tend to use the name Krebs cycle, and so does your textbook. So I'll just continue using the name Krebs cycle. It's called Krebs cycle after Dr. Krebs, who discovered it. In the Krebs cycle, and we have the preparatory steps shown here, the acetyl coenzyme A binds to oxaloacetic acid in the Krebs cycle to make citric acid. And you do not need to know this intermediate of the Krebs cycle, citric acid, which makes this intermediate, which makes that one, which makes this one, which makes that one. And you can notice when we're going from here to here, we actually make some ATP. And then we go to this one and go to that one, and then go back to oxaloacetic acid. Why we call this a cycle is because we're ending up in the place where we began, meaning this cycle will just keep on running as long as you have food that you're digesting and as long as you're alive. It won't ever end unless you're starving and you have nothing to eat, or you die. Each cycle of the Krebs cycle and the preparatory step generates one ATP, shown here, 4 NADH, 1 in the preparatory step, 1 right here, 1 right here, and 1 right there. And FADH2, 1 right here. And then 3 CO2s, 1 here, 1 here, and 1 there. Any questions about any of that? Now understand, the preparatory step in the Krebs cycle will run 2 times for each molecule of glucose, because glucose will, in glycolysis, make 2 pyruvic acids. So the preparatory step in the Krebs cycle can run 2 times per molecule of glucose. So you have to be careful when I'm asking you a question. How many ATP are made by the Krebs cycle? I have to define that by a single cycle of the Krebs cycle, or by 1 pyruvic acid, or will I say how many ATP are made by the Krebs cycle per molecule of glucose? If it's per molecule of glucose, you need to multiply these numbers by 2. Any question about any of that? Let's talk a little bit about the CO2s, and that is glucose is a 6-carbon molecule. There are 3 carbon dioxide made by the preparatory step and the Krebs cycle, but it runs twice. So that means there's 6 CO2 made per molecule of glucose. That means every carbon in glucose is converted into CO2. Any questions about any of that? The importance of that is that every carbon is converted in glucose to CO2. And that's why we breathe out CO2. None of that carbon dioxide came from oxygen. All of it is from the burning of glucose, or the metabolism of glucose in aerobic respiration to make CO2. Any question about any of that? All right, so that's why you have to breathe out CO2. So here we're seeing glycolysis, the preparatory step in the Krebs cycle, and NADH are taking the electrons that ultimately came from glucose. And in the Krebs cycle, they're coming from a molecule that was derived from glucose. And then NADH and FADH2 are taking these electrons to the electron transport chain. In the electron transport chain, we'll then have ATP production kicking into high gear. We have made some ATP by glycolysis and the preparatory step in the Krebs cycle, but only 2 in glycolysis, and only 2 ATP per molecule of glucose in the Krebs cycle. Any question about any of that? All right. So the next step is the electron transport chain. It's sometimes abbreviated ETC. The electron transport chain has a whole bunch of different names. I will only barely use one other name, and that is, it's also called the electron transfer chain, electron transport chain. Those names are similar enough, students don't usually get confused by those names, but it's also called chemiosmosis and oxidative phosphorylation. I don't use those later terms because they're not similar to electron transport chain. The electron transport chain is a series of carrier molecules that are in turn reduced and oxidized as electrons coming from glucose are passed down the chain. Energy is released as the electrons are transferred from the high energy molecules to the lower energy compounds. So each time the electrons are transferred, some of that energy in the electrons are given off, and that's why the cells can perform aerobic respiration at 37 degrees Celsius instead of the temperature you would require to burn glucose, like if you took a blowtorch to it, and that would be like 1,000 degrees. I don't remember if that's 1,000 degrees Fahrenheit or 1,000 degrees Celsius. It doesn't matter. It's quite a bit warmer than 37 degrees Celsius. All right, let's watch a little video on aerobic transportation. If I can get that to run. I'm not sure that's running. There we go.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, 2 mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
All right, any questions about that video? So as I've mentioned, in eukaryotes, the electron transport chain occurs along and through the inner mitochondrial membrane. So let me blow this up. So this membrane containing the electron transport chain, shown here, is in the inner mitochondrial membrane in eukaryotes. In prokaryotes, this electron transport chain is actually in the cell membrane. What happens is NADH and FADH2 bring the electrons coming from glucose, and NADH gives the electron to the first protein in a chain of proteins in the membrane. The electrons move from the first protein to the second, to the third, to the fourth, to the fifth, and then they leave the electron transport chain, and the electrons go to oxygen. So oxygen is said to be the final electron acceptor. Oxygen binds with those electrons into hydrogen ions to make water. And this is why we have to breathe in oxygen. None of that oxygen is converted into CO2. All of that oxygen is converted in the electron transport chain of aerobic respiration into water. Now, humans tend to breathe out a lot of that water that they make in the electron transport chain, but we do retain some of it for our own water needs. Desert animals are very good at retaining that water they make from the electron transport chain. And like desert mice, for example, 60% of the water they get is from the electron transport chain of aerobic respiration. Any question about any of that? So remember, all the oxygen you breathe in is converted into water. None of it is converted into CO2. When the electrons move through some of the proteins in the chain of the electron transport chain, some of the proteins, the first one, this one here, and this one, and that one, take the energy of the electron moving through the protein and then pump hydrogen ions with that energy from this side of the membrane to that side of the membrane. That creates a twofold gradient. That means there's more hydrogen on this side of the membrane than that side, and there's more positive charges on this side than that side. These hydrogen ions are going to want to move from this side of the membrane to that side of the membrane just by the laws of simple diffusion or second law of thermodynamics, meaning they want to move from areas of high concentration to areas of low concentration. The hydrogen ions, come on mouse, cannot move through this lipid bilayer. except at this protein right here. And when the hydrogen ions move from this side of the membrane to this side through this protein, it gives this protein the energy it needs to phosphorylate and make ATP, meaning you add phosphate and ADP to make ATP. The energy for doing this comes from the potential energy that there's more hydrogen over here and more plus charges over here, and they want to move to where there's less plus charges and less hydrogen. And so that's potential energy difference. That potential energy is used to make ATP. Any questions about any of that? All right, let me mention that NADH is a little more efficient at making ATP because NADH gives its electron to the first protein in the electron transport chain. FADH2 is not as efficient because it donates its electrons, come on mouse, to the second protein, missing the first, and the first protein can pump hydrogen from this membrane, side of the membrane to that side of the membrane. Any questions about any of that? All right, let's talk about why this is called the electron transport chain. And that's because electrons are transported from glucose down the carrier molecules and brought to the electron transport chain where they transport from one protein in the membrane to others. It's also called the electron transfer chain for the same reason. The electrons transfer from one protein in the membrane to the next. Now, what else did I want to talk about? Well, if there's something else, I don't remember it. Oh, we could say that the electron transfer chain or electron transport chain has different names, but I don't use those names. This part here specifically is called chemiosmosis, where the hydrogen ions from this side of the membrane move to that side of the membrane. This is also called oxidative phosphorylation because ADP is phosphorylated. Phosphorylation is just when you add another phosphate group, and ADP is phosphorylated to make ATP. Oh, here's another thing I need to say, and that is cells are very efficient at making ATP. Let me see if I can remember. Oh yeah, the cell can convert the energy in glucose, 40% of the energy in glucose, into ATP. So 40% of the energy that is lost by breaking down glucose, 40% of that energy is captured and put into ATP. And you might say, well, 40% doesn't sound very efficient to me. You're losing more than 50% of the energy. But yet, does anyone know how efficient our best engines are? That we, drive our, mostly our, if it's the best engine, it'll be a experimental car. But how do you think our best engines are? How much energy can we get from gasoline and convert that into the energy of motion? Anyone have a guess? No one has a guess? The best engines we have, and normally it's about 20% efficient. So that would be like on a very good car, it's getting good gas mileage. If you have a plastic experimental car, you can get that efficiency up to 25%. So a cell is much more efficient at converting the energy in glucose than any human engine in converting the energy from whatever to whatever. Mainly gasoline engine to the energy of motion. So the cell is very efficient at using that energy. Any question about any of that? All right. In the electron transport chain, ATP molecules are produced by aerobic respiration. In the electron transport chain, we generate 32 to 34 molecules of ATP per molecule of glucose. Prokaryotes are a little more efficient, and they generate 34 molecules of ATP per molecule of glucose. And we're only talking about the electron transport chain at the moment. Let's talk a little bit about the cells, and that is most of our cells only generate 32 molecules of ATP in the electron transport chain. But some of our cells, 3 of our cells, in fact, are a little more efficient, and they generate 34 molecules of ATP per molecule of glucose. Most of our cells, like our skin cells, our nose cells, our muscle cells, our bone cells, only generate 32 molecules of ATP per molecule of glucose, but our heart, our kidney, and our liver are more efficient, and they generate 34 molecules of ATP per molecule of glucose. What do you know about the heart? Which would make it a reason for why it would be more efficient than your skin cells or your muscle cells. What do you know about the heart? Come on, somebody's got to guess. I'm not going to tell you this one.
The heart has to pump the blood through the entire body.
Yeah, is the heart ever resting?
No, never.
No, it's always beating for as long as you're alive. So the heart is always working. Well, guess what? The kidneys are always filtering the blood, recycling the blood, whatever, purifying the blood, pulling out the waste. The liver is also always working. These cells are always working. They never get a rest, time to rest. And your muscle cells right now are probably resting, right? Your brain cells right now are probably resting, except for the one student who gave the comment. And that's why they don't need to be as efficient. But the heart and the kidneys and the liver are always working, so they're more efficient. And they do generate 34 molecules of ATP per molecule of glucose. In aerobic respiration, the final electron acceptor is oxygen, and none of that oxygen is converted into CO2. All of it is converted into water. So here we're looking at the electron transport chain. Actually, this is aerobic respiration. Here's the electron transport chain. We'll talk about it here, where the electrons, which flow from glucose to NADH or FADH2, then they're brought to these proteins in the membrane of the electron transport chain. And then when they transfer from one protein to another, they give off a little bit of energy so that all of the energy in glucose is not released at one time. Because if it was released at one time, the temperature of the cell would be around 1,000 degrees. And it doesn't matter if that's Fahrenheit or Celsius, that would fry the cell. And we do this all at 37 degrees Celsius. Now here it's showing you that the electron transport chain happens in the inner mitochondrial membrane, which is right here, of a eukaryote cell. And the hydrogen ions are pumped into the space between the inner membrane and the outer membrane of the mitochondria. So that protects the cell from these hydrogen ions because they're kept in the space between the outer membrane and the inner membrane of a eukaryote. Any question about that? In the prokaryote, this is happening, the electron transport chain in the cell membrane, and these hydrogen ions are pumped into the cell wall of a bacteria that has a cell wall. Any questions about any of that? All right. I've already talked about that, so I'm not going to talk about it again. I'll just emphasize that the potential energy difference of having all these hydrogen and positive charges on this side of the membrane to that side of the membrane is what allows the cell to make ATP when the hydrogens come through this membrane at this protein. This protein can take phosphate and ADP and make ATP. And this is called chemoosmosis. It's the same as part of the electron transport chain, so I don't use this term. And when you are making ATP, that is phosphorylation, you're adding a phosphate to ADP, and you're having oxidation happening. So it's called oxidative phosphorylation. I don't use that term. I only use the term electron transport chain. So with the ATP made in the electron transport chain and the ATP made in the Krebs cycle and the ATP made in glycolysis, we then have a total accumulation of ATP from aerobic respiration per molecule of glucose in eukaryotes is 36 to 38 ATP, depending on which cell we're talking about. Remember, most eukaryotes only generate 36 molecules of ATP, but a few cells, the heart, the liver, and the kidneys, generate 38. Prokaryotes are a little more efficient, and they generate 38 molecules of ATP per molecule of glucose. The overall summary reaction for aerobic respiration, which you do need to know, is glucose, C6H12O6, plus 6 oxygen atoms, 38 ADP, 38 phosphates, and that's an I for inorganic phosphate, generate 6 carbon dioxide molecules, 6 water molecules, and 38 ATP molecules. Any question about any of that? All right. So this is showing you the steps of aerobic respiration for a prokaryote cell or for the heart, the liver, and the kidneys, where we generate from glycolysis 2 NADH, 2 ATP, and then the preparatory step, the Krebs cycle, make 8 molecules of NADH, 2 molecules of FADH2, and then 6 molecules of CO2, and 2 molecules of ATP. The NADH and the FADH2 carry the electrons ultimately from glucose to the electron transport chain, where we make 34 ATP for a prokaryote cell. And the hydrogens and the electrons from NADH and FADH2 flow to oxygen, which generate water. And altogether, the number of ATP made in prokaryotes is 38 ATP per molecule of glucose. Any question about any of that? All right, so that's it for aerobic respiration. Let's talk briefly about anaerobic respiration. Anaerobic respiration differs from aerobic respiration in that the electrons flowing from glucose do not flow to oxygen. The final electron acceptor is some molecule other than oxygen. It really depends on what form of anaerobic respiration we're talking about. Sometimes the electrons can flow to nitrate. So that would be nitrate anaerobic respiration, but sometimes they flow to sulfate anaerobic respiration. Sometimes they flow to nitrite. You don't need to know that, but that's NO2 with a negative charge. There are many different forms of anaerobic respiration, and as far as I know, they each have a separate electron acceptor. Any question about any of that? In anaerobic respiration, we have glycolysis in the Krebs cycle. So glucose enters glycolysis, and then the pyruvate goes through the preparatory step to enter the Krebs cycle. So all of that's happening. And then we have NADH and FADH2 carrying the electrons to the electron transport chain. However, the protein's chain in the electron transport chain is shorter in anaerobic respiration than aerobic respiration, meaning there's fewer proteins in the chain. And that means the yield of ATP made in anaerobic respiration is less than the yield of ATP made in aerobic respiration. So aerobic respiration is the most efficient. The ATP yield in anaerobic respiration is greater than 2, and the highest would be 36. They're either up to 36 or they're less than 36. So let's say 4 to 36 is the yield of ATP in anaerobic respiration. And the exact yield depends on which form of anaerobic respiration we're performing. And one of the least efficient, it's either nitrate or nitrate. Nitrite or nitrate is one of the least efficient forms of anaerobic respiration. And you don't need to know that, I'm just mentioning it. Because anaerobes make less ATP per molecule of glucose, they tend to grow slower than aerobes. So an anaerobe will tend to grow slower than an aerobe because an aerobe can make more ATP per molecule of glucose. Any question about any of that? All right, that's it for anaerobic respiration. Let me briefly show you one thing. I did mention it, but here we see the electron transport chain of aerobic respiration, and there's fewer proteins in the electron transport chain of anaerobic respiration. Usually you start losing these molecules, but the worst may only have this protein. I'm not sure exactly which one is the worst, but it may only have this protein, meaning the electron transport chain of anaerobic respiration is fewer proteins, a smaller chain of proteins. Where were we? Oh, let's go back. All right, so that's it for anaerobic respiration. Let's talk about fermentation. Fermentation is a separate reaction from aerobic respiration or from respiration, meaning anaerobic respiration. Fermentation does take glucose and it does make pyruvic acid. And then you have the fermentation steps. So first you have glycolysis and fermentation, And then you have the fermentation steps. The fermentation steps are where you convert pyruvic acid to the end product of fermentation, and that's made in either one or two biochemical reactions. So you only have one or two biochemical reactions or one or two enzymes. Any question about any of that? All right. Fermentation does release energy from the oxidation of a molecule such as glucose, although it can be essentially any other food we eat, can go through aerobic respiration or fermentation, but mostly glucose is the molecule which is used. Glycolysis proceeds, as previously discussed, producing two pyruvic acids from glucose, two net gain of ATP, NADH. After glycolysis, we then take the pyruvic acid and attach a fermentation step or two, depending on which fermentation we're talking about. Fermentation does not require oxygen. It can happen in the absence of oxygen or in the presence of oxygen. The final electron acceptor, taking the electrons flowing from glucose, is an organic molecule. So pyruvic acid may take the electron, or it may be the fermentation end product. Pyruvic acid will be converted into the fermentation end product. Along the way, we do generate NADH and ATP here, and you'll note that all of the ATP made in fermentation is the ATP we get out of glycolysis. This NADH is then taken in the fermentation step and used up to convert pyruvic acid to the fermentation end product. It depends on which fermentation reaction we're talking about. It can be either ethanol or it can be lactic acid. We'll talk about those two fermentation reactions. It can be another molecule. We're not going to talk about other fermentation reactions, though. In lactic acid fermentation, it's one of the simpler fermentation steps. You convert the pyruvic acid in one biochemical reaction to lactic acid. Lactic acid fermentation actually happens quite a bit. Most fermented food uses lactic acid fermentation. So it's lactic acid fermentation we use when we're making yogurt, when we're making sauerkraut, when you're making chocolate, when you're making most cheeses like cheddar cheese. Not all cheese, but most cheese. The lactic acid that builds up actually helps preserve the food, why sauerkraut does not easily spoil, why yogurt does not easily spoil. If you don't know, yogurt has a longer half-life than even pasteurized milk, which pasteurized milk is treated to remove the human pathogens, as well as it removes many of the microbes in the milk, but not all of them. The point is yogurt can last longer, and it's because lactic acid acts as a preservative. It does prevent food from spoiling. It doesn't totally prevent it from spoiling. Like you can't leave the yogurt at room temperature for probably longer than a day. I'm not sure how long you can do it, but you certainly wouldn't want to leave it at room temperature for a week. Any question about any of that? All right. Another fermentation reaction you should know is alcoholic fermentation, where pyruvic acid is converted in two steps to ethanol. Ethanol is a 2-carbon molecule. Pyruvic acid is a 3-carbon molecule. So 1 carbon is given off as CO2. And actually in alcoholic fermentation, that CO2 is what gives beer its fizz. Wine is made in an open barrel or an open casket. And so the CO2 is vented to the air. And so you don't get it in the bottle of wine. But if you were to ferment the wine in a closed capped bottle of wine, and that would trap the CO2 in it, you can do that. And we don't call that wine, we call that champagne. It's the same thing, it's just got the CO2 trapped in it. Any question about any of that? Here's a question for you, and that is, in glycolysis, we make NADH, and then that NADH is used in the fermentation product to make the end product of fermentation. Now, why, like for example, our muscle cells, why would our muscle cells use up this NADH and undergo fermentation, which our muscle cells do whenever we're working our muscles really hard, like an Olympic runner or something like that, or you're just running all the time or exercising all the time, you're really working out. And what's happening is the muscles cannot get the oxygen from the blood fast enough. And so the muscles prefer working with aerobic respiration, but if oxygen is not available, the muscle will fall back on fermentation and ferment glucose to lactic acid. And that's why you can feel the muscle burn. That's the lactic acid burning up, building up in your muscle. And the point is the muscle takes this NADH made from glycolysis right here, and then uses it up in the fermentation reaction. Why would the muscle cell use up this NADH rather than just keep it and store it? Because sooner or later, the heart and the lungs are working like mad to give the muscle oxygen, but they can't give enough to it, and so the muscle uses up the oxygen that's there and then engages in fermentation. But why would the muscle cell use up this NADH rather than just store it? Because if it were to store it, it could then use this NADH from the electron transport chain and make a lot more ATP from this NADH than it would from engaging in fermentation. Anyone have a guess? It's actually shown on this graph here. All right, the muscle cell wants to work when there's no oxygen around, so it engages in fermentation to make ATP. As long as the muscle cell has ATP, the muscle can continue contracting, continue working. Occasionally, you might have seen an Olympic athlete, there's usually one during each Olympics, where the Olympic athlete pushes themselves to the limit and their muscles run out of all glucose. Then they cannot engage in fermentation. They make no ATP. When there's no ATP, the muscles stop working. The Olympic athlete usually just collapses because their muscles can't work. The coach then runs out. In the wintertime, they'll cover them with a blanket. I think in the summertime, they run out and they spray them down with water. But the heart and the lungs are working like crazy to give the muscles glucose and oxygen. And when the muscles are rejuvenated with at least some oxygen and some glucose, the Olympic athlete will stand up and then walk across the finish line. I'm sure you've all seen that. Like I said, it usually happens once during each each Olympics. There's usually one athlete that happens to. The point is the muscle wants to engage in fermentation, so it has to use up this NADH to engage in fermentation to make some ATP for the muscles to work. If it were to store the NADH, then there would be no NAD plus because it's made right there in fermentation. And if there's no NAD plus, the muscle cannot engage in fermentation. No fermentation, there's no ATP that's made. No ATP, the muscle can't work. Any questions about any of that? All right, here's looking at some different fermentation reactions. We've only discussed two, so you only need to know lactic acid fermentation and alcoholic fermentation. Like I said, most fermentation reactions are lactic acid fermentation, such as when we make yogurt or cheddar cheese or sauerkraut or chocolate or sausage. Almost everything you can think of that's a fermented food uses lactic acid fermentation. Almost everything. There's a few things that are not. Saccharomyces engages in alcoholic fermentation, and we do use that to make beer and wine. We also use alcoholic fermentation to make leavened bread. That's why we add yeast to our bread to make the bread rise. That's from the CO2 given off to make the bread rise. But that's about it. The only three reactions I can think of that use alcoholic fermentation. And sourdough bread uses both alcoholic fermentation and I think lactic acid fermentation. I'm not sure what the other one is, but my guess is it's lactic acid fermentation. Sourdough bread uses more than one. There is one I'll briefly mention. You don't need to know about it. It's propionic acid fermentation. This is how we make Swiss cheese. Propionic acid fermentation gives off CO2. And you know the holes in Swiss cheese? That's from the production of CO2 in the making of the Swiss cheese. We do have other fermentation reactions, and they're important in industry, like we can use fermentation to make butanol or acetone, succinic acid or acetic acid. Right here, formic acid. One of these two, I'm not sure if it's this one or that one, is used in the making of vinegar. I think it's this one here, acetic acid. Yeah, it's this one. Although I didn't think it was Escherichia or Salmonella that make vinegar. Come on, mouse. Anyways, just a different number of fermentation reactions. Like I said, you don't need to know any of them except for the first two. Here showing you some different fermentation reactions. The industrial commercial use while they're used. Oh, I guess I forgot about this one. And that is ethanolic fermentation or alcoholic fermentation is also used to make fuel. The alcohol that's put in our gasoline is made by alcoholic fermentation. Lactic acid fermentation used in making cheese, yogurt, rye bread, sauerkraut, summer sausage, chocolate, most of the things you can think of. Exception is Swiss cheese, the only thing I can think of that uses this one. And there's acetic acid fermentation. Let me see if that one, Acetobacter, that's right. All right, let's take a look at the summary of aerobic respiration. How are we doing time-wise? Oh, we got a few minutes. Aerobic respiration, comparing it to anaerobic respiration. The difference is Aerobic respiration happens in the presence of oxygen. Oxygen is the final electron acceptor. Anaerobic respiration happens anaerobically. The final electron acceptor is some molecule other than oxygen. Fermentation can happen either with the presence of oxygen or the absence of oxygen. And the final electron acceptor is an organic chemical molecule. We generate 2 ATP in fermentation. In aerobic respiration, for eukaryotes, it's 36 to 38 molecules of ATP, depending on the cell. For prokaryotes, it's always 38 molecules of ATP per molecule of glucose. For anaerobic respiration, it depends on which form of anaerobic respiration we're talking about. Generally, between 4 and 36 molecules of ATP are made by anaerobic respiration. Any questions about any of this? All right, if not, what the heck happened to my slideshow? Oh, for some reason I did the end of the show, but it didn't end the show. All right, let's talk a little bit about lipid and protein catabolism. Glucose is the primary molecule used in, well, actually aerobic respiration, anaerobic respiration, and fermentation, but other carbohydrates can be used. Mostly the other carbohydrates are broken down and converted into glucose. Even if you have like beta galactose as your sugar, that beta galactose will be converted into glucose, and then glucose will be sent down the pathway in the normal way. We can, though, digest other foods, such as lipids and protein, and they can be broken down, and we can make ATP out of them. There's a problem with large molecules, large carbohydrates, large lipids, and large proteins, and that is they cannot enter the cell. They cannot get through the cell membrane. And both our cells and bacteria solve this by secreting extracellular lipases or proteases to digest the large molecule outside of the cell. So the proteases will digest the protein to amino acids, and then the amino acids can be brought across the cell membrane with the transport protein. Lipids will be broken down into glycolysis and fatty acids.
I'm sorry, can I ask you a question?
Yes.
Your slideshow stopped and I'm just wondering if there's more slides to go along with.
I mean,
what
I'm
we're
not
talking
showing
about.
I'm not sharing my screen, sorry.
No, it's okay.
I'm not sure why that happened. Like I said, I tried to end the slideshow.
Yeah,
It didn't work right. I don't know what.
okay, there
All right,
it is.
there it
Thank
is.
you.
Yes. Hopefully this is the only slide in that one. I might have not shown that one, which I meant to. Anyways, thanks for letting me know. Lipids are broken down into glycerol and fatty acids. The glycerol enters the Krebs cycle. The fatty acids are cleaved into 2 carbon chunks, and then those 2 carbon chunks are enter the Krebs cycle. They're converted into acetyl coenzyme A and then enter the Krebs cycle. Amino acids will have the amino group removed, and then they will be converted to enter, depending on which amino acid it is, it can enter anywhere in the aerobic respiration. Most of them enter in the Krebs cycle. There are a few, though, that enter in glycolysis. And I forgot where the other 2 enter. A few enter in glycolysis, a few enter in different steps in the Krebs cycle. So this is trying to show you, let me get this out of the way, proteins digested outside the cell by a protease, making the amino acids. The lipids, which for some reason it's over there, is broken down into glycerol and the fatty acids, and the fatty acids are broken down into 2 carbon chunks, and then it enters the Krebs cycle. So this is trying to show you the metabolism of carbohydrates broken down into glucose, and then lipids broken down into glycerol and fatty acids, glycerol entering the midway through the glycolysis, fatty acids enter the Krebs cycle. Proteins will be broken down with amino acids. You remove the amino group, and some of the amino acids will enter late in glycolysis. Some of them will enter early in the Krebs cycle. Some will enter midway or late in the Krebs cycle. The point is, carbohydrates, lipids, and proteins can all be used as a source of electrons for aerobic respiration or anaerobic respiration, or even for fermentation. The most efficient way of generating energy is, of course, using aerobic respiration. And then the energy in ATP is used for the cell's work, such as allowing the cell to transport a molecule across the membrane, allowing the cell to move, or allowing the cell to make new molecules, anabolism. Most cellular ATP is actually used for making larger biosynthetic molecules. Anabolic and catabolic pathways are actually joined by common intermediates. We call them an amphibolic pathway if the metabolic pathway can function in both directions. An amphibolic pathway bridges the pathways involved in carbohydrate, lipid, and protein, as well as nucleic acid metabolism. So here we're looking at aerobic respiration, and wherever you can see 2 arrows, one going this way, one going that way, it means it is an amphibolic pathway. This doesn't show 2 arrows, but you see this one has an arrow going that way, meaning you can go from this molecule to here, or you can go from this molecule to there, or you can go from this molecule to there. It doesn't matter. The point is, if you eat too much carbohydrates, you all know this, the carbohydrate will be converted into glycerol, and the carbohydrates will be converted into fatty acids, and then you will make lipids, and you'll store down that lipids, right? That's what's going to happen if you eat too much carbohydrates. And we all know that. Okay? The same thing if you were to eat too much proteins, although that's not shown here, but you take the protein, you break it down into the amino acids, and then you can convert the protein into fatty acids and glycerol, and then make the lipids. Now, it is true that if you eat a bunch of lipids, you can digest that, making acetyl coenzyme A and glycerol coming down this way, and then burn the lipid. Most of the time, though, for us couch potatoes, if we eat only lipids, we will actually go reverse and convert that lipid into glucose. And then when we need energy, we'll send glucose down the aerobic respiration pathway the normal way. So most of us couch potatoes will not directly burn a lipid in aerobic respiration. We'll convert the lipid and then go backwards in this pathway to make glucose. The same thing if you were a couch potato like me and you were to eat a bunch of proteins, you convert that protein to amino acids and then go back and make glucose. We might store down the glucose as glycolysis or something like that. But when we need energy, we would then send glucose through the pathway in the normal way to make aerobic respiration. That's not true for an Olympic athlete or a marathon runner. A marathon runner can directly digest lipids in aerobic respiration. It does take the marathon runner 45 minutes into their run before the marathon runner will start digesting lipids. But after 45 minutes into their run, a marathon runner will be digesting lipids in aerobic respiration. So it can happen. It just, the human body doesn't prefer to have it work that way. Meaning, if you're not a marathon runner or an Olympic athlete or something like that, you're not working out a lot, you will convert that lipid into glucose. And then when you need ATP, you burn glucose in the normal way. Any questions about any of that? All right, let me see how we are on the time. Yep, sorry, I'm a little bit past the time here. Let's end it here. And I'm going to be a little bit late in the lab because we only have 6 minutes. But are there any questions?
No, but thank you for explaining everything.
Okay. All right, we're going to stop here and I'll see you in the lab. Let's say about 3 minutes late in the lab.
Okay,
Okay. All right.
thank you.
All right. Bye.